Welcome, everyone. We will get started on the hour. Hello, everyone, and thank you for joining today. Welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, and I sit within Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA within the last few years. We established the seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted on our NOAA Omics website, and there should be a link to the website in the chat now. As participants, you are muted, but feel free to type any questions into the questions for staff box throughout the presentation. At the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Exploring the Deep Sea Coral Communities of the Central Pacific with Genome Skimming and Environmental DNA by Dr. Meredith Everett and Dr. Steve Auscavitch. I'm going to go ahead and share presentation with Meredith while she gets those slides up. Dr. Meredith Everett is a research biologist in the Conservation Biology Division at NOAA's Northwest Fisheries Science Center and on temporary detail as a lead scientist with NOAA's Deep Sea Coral Research and Technology Program. Meredith's genetic work has focused on the applications of omics tools to better understanding deep sea coral communities within the US EEZ, with a particular focus on application and development of eDNA technologies. Meredith has had the opportunity to apply these tools to habitats across the Central and Northeast Pacific, within the Gulf of Mexico, and across the Arctic. Meredith received her bachelor's in science in biological oceanography from the University of Washington and her PhD in marine biology from the University of Miami. Dr. Steve Auscavitch is a postdoctoral researcher in the Department of Biology at Boston University. His research broadly seeks to understand evolutionary and ecological patterns and processes structuring deep sea biodiversity, particularly on seamount environments. His work focuses on identifying and filling knowledge gaps from poorly understood deep sea environments using collection-based approaches, including museum specimens and those collected through exploratory remotely operated vehicle dives. His current projects in the Roentgen lab seek to identify and describe patterns of deep sea coral biodiversity using next-generation DNA sequencing approaches to build genetic reference libraries for the equatorial Central Pacific and surrounding waters. Steve is also an active member on the EV Nautilus Corps of Exploration, where he contributed as a science lead and science data manager on many cruises since 2014. He received his Bachelor's of Science in Marine Science from the University of Connecticut at Avery Point and his Master's in Marine Biology at the University of Maine and his PhD in Biology from Temple University. You can follow his work more on social media at Steve Auscavitch on Twitter. With that, thank you, Meredith and Steve, for presenting today, and I will now pass it off to you.
Thank you, Nicole. So thank you to everyone who's joining us today. And I'd actually like to go ahead and acknowledge our third co-author here, Randy Rowan. She's not helping us present today, but has been critical to getting this work done. So today we're going to talk to you about how we're exploring deep sea coral communities of the Central Pacific with eDNA and genome skimming techniques. And first, I'd like to just kind of introduce us to the whole ecosystem. So both Steve and I work in deep sea coral habitats. Now, when most people think of corals, they think of tropical coral reefs, they think about challenges like coral bleaching, but we work in a very different type of habitat, those in the deep ocean. And so deep sea corals are actually found worldwide in all ocean basins at depths below 200 meters, all the way down to abyssal plain depths. And why we're interested is they're a really important source of biodiversity and biogenic habitat in the deep sea. And they often now, we're finding, occur in areas that overlap with areas that are important to humans for things like fishing and other types of exploitation. And these areas include our study area, the Central Pacific. So I just wanted to introduce our habitat to you. So welcome to the Central Pacific. As you can see from this map, it's a huge geographic area. There are points in here, if you're looking at the globe from space, where you cannot see continental land masses. And it was once thought that this whole area, once you got outside of the kind of nearshore reef ecosystems, was kind of a marine desert. But what we're finding is that particularly around seamounts, there are these really dense, diverse deep sea benthic communities. However, due to the wide geography of this region and just the difficulties of working in the deep sea, which I'm going to talk more about in a minute, despite ongoing efforts over the last decade to better scientifically characterize these communities, we have still thousands of seamounts left to explore in these habitats. So, as I mentioned, seamounts in the open Pacific can be really important hotspots for biodiversity, but we don't know much about the composition of their communities, the level of endemism, so which organisms are unique to specific seamounts versus those that actually span multiple seamount habitats, and the overall population structure of these communities. But we do know that despite their remote location, they are being subject to an increase in anthropogenic pressures. These include fishing pressure. So this central image here, you'll see this is actually trawl scars in a corallium community in the Emperor Seamount chain. So these communities can come within depths that are being impacted by human fisheries. An emerging challenge for these communities is the potential threat of deep sea mineral mining. We don't actually know how mineral mining might affect these communities, but it is a challenge that's going to be taking place in the near future. And then there's climate change challenges. While the deep sea itself can be pretty stable temperature-wise, these communities are all dependent on food that trickles down through the water column to the seafloor to survive. And anything that changes the overall dynamic in the higher levels of the water column can thus have an effect on these communities. So we need to better characterize these deep sea coral habitats so we can better understand how they're affected by these challenges and how we can work to protect them. But working in the deep sea is really challenging. It's challenging just to get there. You're working in the dark. It takes a lot of technology like ROVs or AUVs just to access it. And when we're working with these, we're often doing video transects. We're getting video information of what's on the seafloor. However, one of the challenges there is we can miss things because we just don't see them. You're limited to the field of view of the camera and the light pool. And so you can have really large, important habitat forming species that because they don't occur in your camera image, you'll miss them in your survey. We're also limited in the number of samples we can take. This is both by choice. We don't want to sample so much that we impact a community, but we also only have a limited amount of space on a vessel. And so we have to be really targeted in the samples we take. And these two factors can make it really difficult to overall profile a community and then compare between communities across different seamount chains. So what we're starting to do is apply omics tools to address these challenges. And one important tool that we're starting to use is environmental DNA. Now, just to give a quick overview of what we mean with environmental DNA, in our case, what we're doing is sequencing DNA from cells that we collect directly out of the water column. So we'd go down with an ROV, we take water samples in the habitat itself, we then filter usually up to about 2 liters of water aboard the ship and preserve those samples for processing back in the lab. In the lab, we extract DNA, and then we're applying a process called amplicon sequencing, where we're sequencing a small fragment of the mitochondrial genome, in our case, a gene called mut-S, and then we're amplifying that across all samples, running a bioinformatics pipeline, and what that eventually allows us to do is to get, hopefully, species-level assignments for all of the animals within the local habitat where that sample was taken. So We did a pilot study with this in the Central Pacific. Back in 2018, we were able to collect some samples during an expedition to Papahanaumokuakea, and we just tried this out to see were we able to identify corals in the local area. And what we found was we were getting hits. We were able to sequence the MUTAS gene out of the seawater samples, but we weren't seeing as much diversity in our results as we expect from what we were seeing on the ground in the community. However, these results were based on relatively shallow sequencing depth, so we didn't get that many sequences. We used a smaller sequencing cartridge. And we also had at that point a really limited reference library for species from the Central Pacific. So we had numerous reads that remained unassigned. So we realized we needed more samples, deeper sequencing, and more overall genomic resources. And that's kind of what this project is about, because eDNA is only as good as your reference library. And at this point, I'm going to turn things over to my colleague Steve to talk about that.
Thanks, Meredith, and thanks, Nicole. So as Meredith mentioned, our eDNA tools are really only as good as the reference library that we can apply our eDNA amplicons to. So part of my work has been focused on trying to generate a genetic reference library useful in this project that is taxonomically and phylogenetically broad, but also allows us to get as close as possible to species specificity as we try to identify these amplicons that come up in our eDNA sequencing. So just to give you a perspective, this image here is from a cruise this past summer in Johnston Atoll aboard the EV Nautilus, where we observed high density communities like this with multiple different families of deepwater octocorals present. Most of what I'm going to talk about for the next few slides is going to be octocoral specific, since they are the most diverse coral group in the bathyal depth range in this region, sometimes accounting for over 60% of the species that you might find on a typical seamount. But also there's a lot of taxonomic diversity that we're still trying to tease apart. Next slide, please. So as Meredith mentioned, eDNA tools can be really powerful, but having those reference libraries is almost as important as obtaining the eDNA samples themselves. So one of the challenges we sought to address is trying to determine if we can get species specificity in these studies from the amplicons that we were sequencing, and using any available specimens, both from those that were collected recently aboard exploration cruises, as well as museum archives, which I'll talk about in a few minutes. And one of the challenges in designing a library, well, first is obtaining the samples that from the geographic regions and the taxonomic groups that are important for your eDNA target species, but also identifying genetic markers which offer a broad taxonomic signal and potentially species specificity, so you don't exclude particular species based on choice of marker. So one of the other approaches that we find very attractive about eDNA in this type of environment is particularly where we have culturally or significant sampling areas like national marine monuments or national marine sanctuaries. It allows us to minimize our sampling to vouchers, small clippings of corals, without having to take a large specimen and relying on samples that are already in archive. Next, please. So just to introduce you to some of the species that we might be talking about here, these are all deepwater octocorals from across the Central Pacific region. The majority of these are actually from the Northern Phoenix Archipelago, the US Pacific Remote Islands Marine National Monument unit around Howland and Baker Island. And a lot of these species occur between maybe 200 down to perhaps 2,500 meters or 3,000 meters in some cases. But in many of these environments, there's precious few collections to date, and even using morphology alone, it's still hard to identify some to species. And in fact, a lot of the taxonomic work that's been done in this region dates back to the early 20th century, with recently, in the past 10 years, some renewed interest coming back and focusing on identifying species, but still the critical component of the genetic infrastructure for these species is lacking. And so that's one of the things that we can address using this technique that I'm about to talk about. Next, please. So we identified genome skimming, a tool that I'll discuss on the next slide, as a way to sequence a lot of samples from a broad range of taxonomic groups, a broad range of geographic areas, a broad range of depths throughout the region, and generate large amounts of genomic data to use to create this reference library. Many of the samples that I'm going to talk about today came from various different exploration cruises that were parts of multi-year, multi-ship campaigns from the NOAA ship Okeanos Explorer and the Capstone program from 2015 to 2017, as well as Ocean Exploration Trust in this area from 2018 to the present, as well as the Schmidt Ocean Institute, where 2 cruises were conducted in the Phoenix Archipelago in 2017 and in 2021, providing a really large amount of samples for us to really get a large breadth and depth range for this library. On the left-hand side, you can see a broad depth swath of the samples that we've collected for the vouchers in our library, and it really targets 2 important aspects of this area. The mid-bathyal zone, so between roughly 1,000 and maybe 2,500 meters, which is a deep water octocoral diversity maximum in the deep sea in this part of the ocean. And it's also coincident with a lot of the summits of these seamounts, which are potentially mining targets, these large flat-topped table mounts, for example. And so we really capture a lot of the diversity and potential areas that could be targeted for anthropogenic impact in the future. The map on the right-hand side just shows the limited number of samples that I'm going to present in the library that we've designed for today, but it's expanding over the next few months. Next, please. The other aspect I wanted to talk about was using samples and specimens already in museum collections. Ocean exploration is fantastic. We can get a lot of new samples, but there's already so many important specimens that are really useful, type specimens that have not yet been sequenced, for example, that we can incorporate in these types of studies that provides a multiplier of the effort that we're trying to accomplish with this genomic work. For example, it allows us to generate genomic sequence data for type specimens and also improving broad taxonomic coverage for phylogenetic studies, which are greatly needed in this area, and this genetic data is quite limited. So what I'm going to talk about today are around 40% of our tissue vouchers in the library are already in museum repositories, primarily at the Museum of Comparative Zoology, as well as the National Museum of Natural History at the Smithsonian Institution. But we're still continuing to deposit and accession these samples so that all of these materials are available, and it's expected we'll have everything accessioned by the start of next year. Next, please. So I mentioned we identified about 300 specimens, 300 collections that we applied this genome skimming methodology to. So our sequence library prep was started with DNA extractions using a plain old QIAGEN DNAZ blood and tissue kit. We extracted and sheared the DNA to the target size required by our library prep kit, which was the NAB Next Ultra 2 library prep kit. And we aimed to do about 192 samples on this first genome skimming run, which we were able to then sequence on an Illumina NovaSeq, and the results of that generated 3.2 billion reads, which were paired 150 base pair fragments of DNA. Using these large data sets, usually around 10 to 25 million reads per sample, with a majority comprising nuclear DNA material, we are able to generate the mitochondrial genes that were really important for our eDNA work. And so we assembled these mitogenomes based on references that were already available in GenBank, for example, to species that were either within the same genus or in the same family. And so this resulted in 171 mitogenome assemblies, and from that we were able to extract 167 of the target eDNA gene sequences that we are able to then apply to our reference library and eDNA amplicons. We also had an additional set of partial mute-S sequences from some previous work that I had conducted in the Phoenix Archipelago, and we added those 88 samples and species to the library. We're also continuing to process another set of genome skimming data with another 88 deepwater coral species, using primarily museum collections, but also some more recent collections from cruises last year aboard the EV Nautilus. And so on the right-hand side, you can see one of the draft assemblies for Solembulula monocephalus, a species that had seldom been observed in the North Pacific or even the Central Pacific, and specimens were really only available in formalin, but we were able to generate a draft mitogenome for this species, which allows us to better understand, for example, its relationship to the same species that may be in other ocean basins. So that's one example of one of the products of this work. Next, please. So here's a summary of kind of the phylogenetic diversity and how many species we identified using the genome skimming run we initially did, plus the additional partial mute-S sequences. From all of those sequences, we found about 255 total sequences. We worked on this data set in Genius, and I used one of their tools to identify potential molecular OTUs, operational taxonomic units, 177 possible species, but it's likely that that there might be some cryptic diversity in our phylogeny, since there's some really different species that are quite similar morphologically, but also have a very different molecular distances from one another. Despite this, we were able to identify these 255 sequences primarily from some of the more speciose families like the Paramecias, Chrysogorgians, and Primnoas, which are among the highest diversity families in these seamounts, but also Corallias, the precious corals, the bamboo corals, Keratoisidids, and other minor families. But this work is primarily just to show the kind of breadth of species diversity that you might find within these families, but also the relative diversity from a sampling of the species you might find in a typical seamount environment in a small geographic area in the Central Pacific. Next, please. What was there was also some interesting downstream effects from doing this initial survey and initial construction of the library. We generated some molecular data for many recently described species, about 12 new recently described species from about the past 5 years that were identified using morphology, and we were able to find some molecular support using some of the genetic and genomic data that we obtained from these collections. Which includes holotypes, which is quite critical for those who are interested in addressing taxonomic questions. In addition, from the species that we were able to sequence, we found and identified some new species candidates from some quite remote areas. For example, in the lower right-hand corner, the new species of Meracides from about 388 meters depth, so sub-mesophotic upper bathyal, from Jarvis Island, which is quite remote, that I'm currently working on. In addition, all of the mitochondrial genome assemblies, 171 partial or complete mitogenomes, are really additional benefits because they allow us to understand the octocoral evolutionary patterns of diversity, biogeography, and also systematic diversity. So all of these tools, once they're made available, can be used quite broadly to address a large range of questions, and not just the aspects of eDNA that we're most interested in. I will turn it back over to Meredith for the next slide.
All right, thank you, Steve. So now that we've generated all of this novel molecular data, we need to incorporate it back into our eDNA pipeline to see if we can get better molecular identifications. So part 2 of this is bringing this back to eDNA and what we're doing with that so far. So while a lot of these tissue samples have been collected, we've also been involved in collecting eDNA samples from across the Central Pacific Basin over the last 4 years. So from 2018 to 22, with a little break for COVID there in the middle, more than 10 cruises to the Pacific Remote Islands Marine National Monument, Papahanaumokuakea, the Phoenix Islands, and American Samoa have collected more than 277 eDNA samples via ROVs. And additional samples have been collected in 2023, and particularly in select regions like Howland Baker, where we were sample deficient in the original collections. We're going to be incorporating the 2023 samples as both time and our resources allow as well. So we also really needed to build a better eDNA database. Our original database in that pilot study was based on the limited sequence resources that were available in NCBI at the time, and we used an RxML-based pipeline. So in the meantime, we've been updating our eDNA reference library and also our data analysis pipeline. The data analysis we're now doing is through the QIIME2 pipeline, and we've used a custom pipeline and an algorithm called Rescript to actually pull all of the available mute-S sequences from NCBI. So when we started with that, there were about 4,000 sequences in there, and once we reduce that down to only the unique sequences or unique taxa, we get about 2,500 sequences. We also pulled all the taxonomic information associated with those sequences. However, NCBI does not always keep their taxonomy up to date. So we've gone through the effort of converting the NCBI taxonomic format to the WORMS, which is the World Ocean database of species, and used their taxonomic designations for all these species because that octocoral database is kept very, very up to date. And we're also incorporating the new sequences from genome skimming. And so we're incorporating, even if we don't have species names for these, we're giving them kind of initial morphological and phylogenomic assignments based on comparisons to the most currently available phylogeny for octocorals. But I'd like to acknowledge that we are expecting to get some higher level species assignments as some of our taxonomic material is currently still undescribed. And this is actually a problem across the octocorals in studies in addition to ours. Octocoral taxonomy is very much in flux right now, and that's presenting a bit of a challenge for us as we work through this project. So our initial database, we've gone through now actually 2 rounds of updating, and we are currently involved in using some of these original pilot samples to test the performance of our database and to continue to add additional resources. So as of yesterday, actually, the sequence database contains about 2,300 mute-S sequences from across the octocoral tree. And we're going to be adding those additional 88 mitogenome sequences in the coming weeks. And then we're also going through and working to refine the database structure to maximize the information we can get out of it. And that's what a little bit more what I'm going to talk about now. So after we'd done the initial database reconstruction, we revisited the pilot samples. So we re-amplified them in the lab, resequenced them, and sequenced them to a much deeper depth than we did in the pilot round, and then ran these samples through the updated pipeline using our new QIIME2 database. And so I'd like to remind you that when we first saw these, we really didn't see that much identifiable sequence diversity. So what does it look like now when we've updated our database? So what we found in a subset of 10 samples that we looked at was that we could detect 92 unique amplicon sequence variants, or ASVs. And we could assign those to primarily family or genus level. And what you can see here is these are those 10 samples, and this is the identifications we can see. And don't worry too much about the small text here. What you should know is we're still seeing a lot of bamboo corals, not surprising in these samples, but we were a little concerned that we weren't really seeing quite the level of sequence diversity that we would expect. So we started to dig a little deeper into these samples. And it turns out that there is actually more diversity than this figure would suggest. What we're dealing with here is a quirk of the way QIIME2 deals with data. It's actually been tuned for dealing with data sets that are much more broad, looking, say, across all mitochondrial or at really broad 16S sequences from microbes. And so family level for that is not surprising, but we want species level. And we also are dealing with ongoing issues in the underlying taxonomy. So for instance, this first column here, within this group that's just been identified as Characterize, so just in the family, there are 12 unique amplicon sequence variants just in this group, and they are unique enough that they are probably 12 different species of bamboo coral. So I took a little bit of a deeper dive into especially the bamboo corals, because we know, working in the deep sea coral community, that that is a taxonomic group that is particularly problematic, but there has been a lot of molecular work applied to that group in the recent past. So our database contains 201 Characterize sequences. However, when we took a deep dive into them, and that's this up on your upper right here, this alignment you can see is just a subset of these, what we find is that the way we built our database, we actually ended up keeping sequences that were identical to one another but had slightly different names. And it turns out that this is not actually that informative for the bamboo corals. We need to remove some of these sequences that have overlapping sequences with unclear nomenclature. So one thing that's gone on in the bamboo corals is there have been several recent genomic studies that have basically shown that with multiple types of molecular markers, you can actually genetically cluster bamboo corals into these letter-clade families here. This is a figure from the recent Morrissey et al. Paper that did just that. And so we can actually look in some of our sequences and find that we do have these letter-clade systems. So all of these sequences from our database are actually here. This is an example from the C1 clade. And so we are getting better information now if we take the time to go back and do a second level curation of our database and really make sure that what we are pulling and including in the database, we can assign to these letter clade groups, and make sure that the samples that we're including have the best taxonomic information possible. And I would actually say that for all groups that are looking to do environmental DNA sequencing, if you are interested in a specific subgroup, so not necessarily looking across, say, all eukaryotes, but you're interested in just, say, rockfish or deep sea coral, I would really promote people to take the time and effort it will take to put together a really well curated database with really well supported taxonomy, because we are starting to see much better assignments the more we can improve the taxonomy of the underlying database. So right now, the stage we're at is actually going through another pass of curation on our database to remove these overlapping sequences and make sure we have high confidence, as high a confidence as is possible in octocorals in the taxonomy of each of the specimens that we include. All of that being said, even with this underlying taxonomic uncertainty, we can start to apply our output to other types of ecological analysis. So this is a Bray-Curtis analysis comparing the 4 different seamounts at which those samples were collected. And what we can see is we can really start to already tease apart different seamount communities and find out what the environmental and local drivers are that might be affecting the diversity of species within each of these communities. So you can see here that we have this one community down here in green from seamount number 7 that is very different and has much less overlap than these other 2. In this case, that's a depth-driven difference. These samples were collected at an overall shallower depth than these other seamount communities. And this is the kind of application that we're going to be taking our environmental DNA data and using to characterize and see if we can figure out what the biogeographic boundaries between these communities in the Central Pacific are. So, wrapping it all up, we're continuing to expand our DNA mute-S voucher collection. We're doing additional genome skimming, and we're continuing to refine our reference database. Right now, we're actually also working on completing the library preparation for all of those eDNA samples so that we can apply our complete database to really better characterizing the deep sea coral communities of the Central Pacific. So we'd like to thank the group, our funders, the Ocean Exploration Trust, and the Deep Sea Coral Program, and the Ocean Exploration Trust. None of this work would be possible without any of them. And then open it up for questions. So thanks again.
Meredith, Steve, thank you so much for this great presentation today. We don't have any questions yet, but I do have a general question that relates a little bit more to the beginning of your presentation. There was this idea to use these findings to better characterize deep sea coral environments. And I'm wondering if there's any way, as we see shifts in shallower coral environments and shallower depths, if you would see a possibility of using these types of databases for looking at these types of changes, but within within shallower depths. I guess really, I guess my question here is, do you think this data can be used to inform changes within environments outside of deep sea corals? Does that make sense?
Yes. So can we use environmental DNA data to track environmental changes that are, and community composition changes that are affecting, say, shallow water coral habitats? We can. It's mostly the barriers are collecting the samples and collecting them over time that will allow you to monitor changes. And then again, having the reference database tools. But environmental DNA has really exploded kind of onto the ecological and omics scene, and it is being used for that kind of work at this point. And you don't have to do it through metabarcoding, which is what we're doing. So if you're really interested in knowing whether one coral species is still present in a community or is successfully spreading to a different location, that say if you're doing habitat restoration, are you getting a coral species that you'd like to start growing there? You could use other techniques like quantitative PCR to monitor specific species at much higher resolution.
Awesome. Thank you for that, Meredith. We do have a few questions coming in now, so I'll go ahead and read them off. This question is from Sean, who asks, have you thought about using phylogeny to correct taxonomic issues?
Yes.
I can address that one a little bit, and I'll toss it over to Meredith. Yeah, definitely. So one of the things that's a sub goal of this project is to improve our understanding of some of the taxonomic relationships among species, not just using the morphology where many of these species were described with, from this area, sometimes, over 100 years ago, but also trying to use their phylogenetic relationships now to clarify and reorganize their the species relationships within a genus or or genera within a family. So this is definitely one of the goals of the project, and I'll let Meredith take it over, add to it.
Yeah, I was going to say from the eDNA perspective, while I'm still going to be using the QIIME2 pipeline, I'm also actually going to bring back that RAXML-based pipeline and look at those two side by side so that we're getting not just the Bayesian clustering, but also able to see where our unique sequences are falling out in a more phylogenetic context, because that does give us a little bit more information than just characterize today, and allows us to do things like clade to the D clade or the C clade for things like the bamboo corals. So going forward, I'm going to be using a bit of a hybrid approach, bringing back that phylogenetic information as well. And we're really excited. A lot of sequencing is going on in the deep sea coral community. The Smithsonian is putting in a huge effort. So we're expecting more and more resources to be available using these techniques.
Yeah. Thanks, Steve, and thanks, Meredith. Our next question comes from Damien, who asks, with such a small custom reference database, have you compared your QIIME2 pipeline to the local BLAST-in from NCBI in order to get better species resolution?
So BLAST does not actually work great for octocorals. So QIIME2 and its background Bayesian algorithm does actually a better job at species assignments with the database. The problem with a lot of octocorals is that you can have differences of just a couple nucleotides between species. And so the issue where we're not seeing the diversity right now is actually more of a naming issue, where we have several underlying sequences in our database that we got from NCBI that are probably actually the same species, but are not currently being called the same species because they were deposited in NCBI just as species X or Keratoisididae themselves. So we're working right now to resolve that and maybe narrow out some of the sequences that just don't have good taxonomic information associated with them.
Our next question comes from Heather, who asks, is this kind of work more or less advanced in other US regions or countries?
Steve, do you want to tackle that on the genome skimming side, and I'll tackle that on the eDNA side?
Yeah, sir, I'll address a couple of points. You know, I'm not completely certain about the applications of genome skimming in the broader world, but I know that there's a lot of interest in the products of something like genome skimming. It's far more cost-effective and cheaper if you have access to the sequencing equipment or sequencing facilities to do it than perhaps generating individual barcodes by Sanger. You get a lot more richness of data. So in that way, it's more efficient, but it also is more restrictive because of access to, like I mentioned, the sequencing technologies. However, the products are already being, the genetic sequence databases are already being sought, you know, far afield. For example, if individuals are doing work in areas that are currently being evaluated for deep sea mining impacts, like in the Clarion-Clipperton zone, this database is directly applicable to that kind of work. So as we continue to develop this database and include new samples, I would encourage folks to use or consider genome skimming and the cost benefits of it versus individual you know, Sanger barcoding to address their questions.
And in terms of the applicability to eDNA, I would say the challenge in this geographic region of the Pacific is that it's undersampled compared to other regions, say within US waters. So we have large collections of specimens, largely coming out of things like the trawl fisheries for things like the US West Coast and Alaska. And over time, more sequencing effort prior to this study had been put into those collections. So if you're working, say, in the Gulf of Mexico, there were already a lot of sequence resources available, which is not to say that genome skimming is not needed in those regions to clarify taxonomy and do things, but going into this project, just the genomic resources available for species from the Central Pacific were very, very low. And so a big part of this effort is just getting those resources available to the community.
Thank you both. Our next question is from Sebastian, who asks, why did you pick mut-S as the marker gene?
So in octocorals, mut-S for using a mitochondrial gene, and right now eDNA is at a spot where it's much more successful to use a mitochondrial marker than some of the nuclear markers, just because mitochondria are more abundant in the cells, so there tends to be just more of that genomic material available in eDNA samples. And within octocorals, mut-S is an octocoral specific sequence, and what it is actually a DNA repair gene. And it's a functional DNA repair gene. And so octocorals tend to have overall less diversity in their mitochondrial genome compared to other eukaryotic taxa. And what this means is a marker like CO1 is really not able to distinguish species in octocorals, but for many groups, mut-S can. And so right now, it's our best available marker for distinguishing species in the Octocorallia.
Thank you, Meredith. We have lots of questions coming in, so we're going to try to get to as many as possible. This one is from Mark. Do you know if the primer mismatches differ among species? If so, do you think that might affect sequence proportions?
So for the eDNA, the mut-S primer we've designed and published is designed around a highly conserved region of the mut-S gene. We did identify, after the original primer design, one area where there were mismatches in one family group, and we designed a second primer to account for that. And when we run our eDNA, we're actually running a blend of the primers. And so right now, we don't think we're seeing any primer bias in our amplification with that blend, but it is something we try to test for. So in each of the eDNA runs I do, I actually usually run a mock community sample where we take some of the genomic DNA from specimens collected in the same regions, make a mock community out of that of known proportions, and compare that to the kind of sequence distribution we're getting, and to make sure we're not seeing primer bias. But it is something we try to keep aware of.
Thanks, Meredith. Our next question comes from Allison, who asks, could the depth of the read and thus the amount of sequences obtained be impacted by the size of filter, i.e. 0.2020 or 0.22 or 0.45 versus 0.45?
Possibly. That's actually something we haven't done a lot of testing on.
Great. Our next question comes from Zachary, who says, wonderful talk. Really exciting work, clearly from your work curating reference databases, particular taxonomic assignments of sequences improves our ability to characterize deep sea corals. How should we as a community be organizing to curate these databases? Do you think this needs to be done on a group by group basis, or could there be an attributor or trusted data broker to address these issues?
I'll give a couple of points, and then maybe Meredith can fill in also. I think generally I will say and preface the comments with that we can never sample too much, particularly when we're looking at differences or small differences in genetic code. So obviously sampling and sequencing more specimens is always helpful, particularly from these areas that represent a very large geographic area. There may be smaller cryptic species, for example, from region to region. But data-wise, yeah, I think having a database that is easily referenceable, whether you're using databases that are already present in NCBI or archives from NCBI, I think having a good curated reference database is really useful and critical and hard work, honestly, to then just pulling things off of NCBI, at random, where there's maybe good quality control, but maybe not specific to the questions or you're trying to pose for your own work or research. Meredith, you have any comments?
Yeah, I think one thing I've learned for this is that, yeah, the curation of the database, as much work as it is really important. It does depend a little the scale, like the ecological scale you're looking at. So if we're looking at, say, across all eukaryotes, the markers that easily amplify all eukaryotes, I really wouldn't be expecting to get any deeper than probably genus level identifications anyway. And so at that scale, some of this is going to be just statistical noise. But if you're really interested in just octocorals or just rockfish, I think having a really well curated database and one that's tied to type specimens whenever possible, or tied to, really well curated, well identified museum specimens. True vouchers is going to end up being really important for being able to get clear species level designations. And yes, it's a lot of work to curate a database like that. But I think it's work that's worthwhile. And so I would encourage the community as a whole to start thinking about, should we be having teams just like the worms database has specific taxonomic teams for making sure the taxonomy stays update, to say, we're a genomic community that works primarily in this taxonomic group. We're associated with museums, with government agencies, with universities. Do we need to be workshopping together to create shared, well-curated databases that can then be combined for larger efforts looking across all eukaryotes? But I think database curation is going to be increasingly important for this type of work.
Thank you. Our next question comes from Kelly, who asks, would a Crux platform with Anacapa be helpful for utilizing the custom database?
Possibly. I would like to try that compared side by side with the chime, and just see if we're getting, what the level of curation we're getting out of each of those is.
Awesome. Our next question asks, hello, thank you for this webinar. Are you obtaining a comparable data, obtaining a comparable data set with eDNA analysis with the deep sea coral diversity that you see in ROV images taken at the same sampling site?
I would say we're not quite to that level of analysis yet, but that is eventually the goal, to compare those side by side. Any additional thoughts, Steve?
Yeah, I will say that there has been a lot of really good work since we have been working alongside groups like the Ocean Exploration Trust, and also have a couple of cruises on the RV Falkor. We've been doing a lot of video annotation of the species, at least as fine as we can identify them from video, and using informed using collections. We've been annotating these videos so it provides us some aspect of diversity that we can then go back to the eDNA samples and see how many amplicon sequences match to species for those particular samples. So that data is there. It hasn't been worked up yet, but we're starting to accumulate a good mass of those types of samples. But it's definitely on our to-do list, and I'm personally motivated to take care of that myself.
Thanks, Steve. Thanks, Meredith. Our next question is from Genevieve, who says, thank you for the awesome presentation. This is essential or exemplary work you're doing for deep sea coral species detection with sequencing for reference specimen. Do you think that the DNA extraction process may limit the detection of some coral species, i.e. due to high concentration of calcium in the area? We've had such problems in other marine environments. Perhaps the problem may not only be with the reference library.
It's something possibly worth looking into. So the way we are extracting the environmental DNA samples, however, is through phenol chloroform extractions, as slow and painful as that is to do. And in some side-by-side tests we've done with eDNA samples from other environments, the quality and quantity of the DNA we get by doing those extractions is higher than with comparable methods. When we do the amplicon amplification reaction, I do actually test for inhibition. But it's worth thinking about and looking about and maybe comparing between a couple different environments or habitats to see if there's any specific pattern if we start to see things like missing species in this going forward.
Thank you. Still a few more questions coming in. This one is from Kelly, who asks, could it be that longer reads are needed for species ID?
Yes, and potentially secondary markers. So we do know that while mute S is the most commonly used kind of barcode for octocorals. We know it's not perfect for all families. And in fact, there's one family that has lost mute S. So secondary markers may be needed, or longer reads.
Yeah, and I'll add too that the genome skimming methodology is a great gap fill for this type of necessary data, because you can get a very large amount of either molecular or nuclear data. You have a lot more flexibility over how you apply your different markers. And so you don't have to go back and amplify individual or smaller segments of DNA using Sanger. So it's a really applicable and flexible tool for this type of work.
Awesome. We have 2 more questions today. One by Jeff, who says, thanks for the great presentation. There was so much work to curate the sequences into the reference library. Is the library specific to Pacific corals, or does it include Atlantic deep sea corals as well?
It includes Atlantic as well, although all the new stuff is Pacific.
And there are quite a number of cosmopolitan species. So Atlantic, Pacific, Indian Ocean. There are quite a number of cosmopolitan species that have been sequenced and are present in this library. So that's applicable too.
Yeah, the current iteration of the reference database is basically for this project is our new sequences plus everything that's available in NCBI. And right now I'm kind of operating by about every 6 weeks. I download anything that's been added to NCBI and get that into the reference database.
Questions here, sorry, misspoke before. Thanks so much, or this question is from Roxanne, who says, thanks so much for the great talk. Do you think the volume of water you're sampling for eDNA is sufficient to capture the full coral diversity in the area? Do you think more water would be useful?
Possibly. So we originally were only doing 1 liter. We've since increased to 2 liters for some of these samples, and are going with 2 liters going forward. Because we are in these deep kind of oligotrophic ecosystems, we don't need as much water as you would need for, say, a tropical coral reef, because there just are not numerically as many species around. More water is always better, but we are also dealing with the limitations and capabilities of ships and the sampling teams on those ships to be able to process the water. So we try to balance those 2 things out.
Alrighty, last one for today is by Arliss, who comments, great talk, thank you. I assume this is probably still a ways into the future. I'm curious what the prospects are for quantifying abundance, density, and biomass, etc., from eDNA in the deep sea.
Stay tuned. I have some other data that I'm hoping to work with some local modelers at Northwest Center here to try out exactly that, and then hopefully we'll be able to do the same thing with these data as well.
Awesome. Thank you so much, Meredith and Steve, for your time today. And thank you, everybody, for joining. We are so grateful to have you here. Our next presentation, our next seminar will be November 8th, and an invite for that seminar will go out later this week. Thank you so much for everybody for attending, especially our panelists, for their great presentation today. Thanks, everyone.